All right, now we're going to look at the activation of these specific pancreatic proteases that are responsible for the further enhancement of digestion. All right, so we begin with digestion at the top here. This is your stomach. So the stomach is responsible for breaking down nutrients further, and after it's done, it passes all of this new material, now this is known as chyme, through the pyloric sphincter, and now it enters the small intestine. So we have our small intestine here. And what is the name of the uppermost region of the small intestine? This is called the duodenum. And in the duodenum, this is where we see majority of digestion and absorption take place. Also here you have your pancreas. So your pancreas is responsible for, when we look at the exocrine function of it, is the creation of these digestive enzymes we call zymogens. So let's write that down. So these digestive enzymes that are inactive, we call them zymogens. And so they need to be stimulated in order to be released into the duodenum. So in the small intestines, there are these specific cell types that we learned before called the enteroendocrine cells. The enteroendocrine cells secrete a hormone into the bloodstream called CCK. So choleocyclokinase. Choleocyclokinase's effect on the pancreas is that it stimulates the acinar cells. So the acinar cells, these cells are responsible for the production of these zymogens, to produce more zymogens and also transport it into the duodenum. And so now in the duodenum, here you have all of these inactive proteins. So this is important. The reason why it's important is because now you need to find a way to activate these proteins, these zymogens, these proteases, because if they don't become active, they're absolutely non-functional, so there's no point of even bringing them in the first place. So in the membrane region of the duodenum, there are these proteins that are being secreted, specific enzymes called enteropeptidases. So these specific enzymes called enteropeptidase. And the function of the enteropeptidase is to create this cascade of cleavages in order to activate these specific proteases. So there are multiple proteases that are inactive in the form of zymogen that enter the duodenum. However, the one that becomes the rate-determining enzyme is trypsinogen. So trypsinogen comes in from the pancreas, so there's all these different proteases that come in here. You got your chylotrypsin, trypsinogens, you have your these procarboxylases. So the main one we want to look at is trypsinogen. So trypsinogen is this peptidase that's inactive. And so with the help of this enteropeptidase, this enzyme helps cleave trypsin into an active form. Now trypsinogen has become trypsin. And this itself is a protease. Trypsin's function is not necessarily in the way of actually breaking down proteins, but it also plays a big role in activating the remaining proteases that are in the zymogen form, in the inactive form. So two proteins, chymotrypsinogen and prodecarboxylase. So again, remember, if you hear the word inogen or pro, this is talking about an enzyme that is not active, but if it becomes cleaved, it becomes active. So decarboxyl and this enzymes. So if you have to understand something about these specific proteases, when we look at fats and carbohydrates, the enzymes that are usually broken are very simple, and what I mean by simple is there's only usually one or two different types. And each metabolite that you get is used for the same physiological process. Amino acids, how many amino acids do we have? We have about 20 amino acids, 10 essential, 10 non-essential. So 10 that we need, 10 that our body can create ourselves. And not only are these proteins or these amino acids important for the building block of proteins, right? So building block of our bodies, but each amino acid actually plays a selective role of different physiological processes. For example, glutamine is a great example of an amino acid that carries the ammoniums that are one of the byproducts of certain kinds of protein synthesis, proteins have been broken down. So you need selective enzymes to break down selective peptide bonds in order to have these specific amino acids for these physiological processes. And so BERT20 requires a bundle of different types of proteases. That's why we have, for example, these are just 3 examples of these proteases. There's much, much more, okay? Now, when we look at this example here, this trypsin comes in here and actually cleaves these 2 enzymes into active proteases. So if I were to draw this here, just for the sake of space, this trypsin comes in and cleaves both the chymotrypsin and the procarboxylase to give you 2 separate enzymes now. So you're going to have these chymotrypsins, again, another protease that's active. So chymotrypsinogen that gets cleaved by trypsin to give you chymotrypsin. And now this other enzyme here is called the decarboxylase. And so this is another enzyme that was inactive, an inactive enzyme also, zymogen, and now it's active. Now it can actually go in and cleave any kind of physiological processes that takes place regarding the protein bonds, these peptide bonds.